Hello. My name is Oscar. Thank you so much for stopping by my poster. I would like to share with you about a new analytical method we have been developing to study gene expression within tissues. Our new method allows to detect gradients of gene expression at the contact zones, or interfaces, of two features within a tissue sample. This is of crucial importance to understand how cells interact spatially within the tissues. It is also a common question during the detection of drug targets for diseases such as cancer and neurodegenerative disorders. As an example, our method helped us find that genes involved in the progression of diseases such as Alzheimer's, Parkinson's, and Huntington's are active in the interface of the white and gray matter of the brain. This finding warrants further research to test for therapies that target those genes in the white-gray matter interface. These results can be seen in Table 1. Our method relies on the use of spatial transcriptomics technologies that sample gene expression at single-cell or near single-cell level. Those technologies quantify the amount of activity of genes, or expression, and the 2D location where that gene expression is occurring. Studies addressing gene expression at the interface of two tissue features, or tissue niches, often rely on the detection of the position of interfaces via clustering. This may introduce biases because a cluster may not capture the gradients of gene expression, or new method uses correlative analysis to tackle that challenge. As mentioned before, we tested our method using a brain cortex dataset generated by Maynard et al. The dataset was generated using the 10x Visium spatial transcriptomics platform. This platform measures gene expression at approximately 5,000 locations, separated each by 100 microns, as seen here in Figure 1. Our method calculates Euclidean distances from each of those locations, which we call spots, to the spots located within a reference tissue niche, as seen here in Figure 3. In our case, the reference tissue niche was the white matter area defined by the authors of the brain dataset. We show the location of the reference white matter region in Figure 2 for each of the 3 samples we included in this study. For each spot, the minimum distance is obtained among all the spot-to-spot distances. Then, Spearman's correlation coefficients are calculated between the Euclidean distances are correlated with the expression of each gene. We also applied a limit to Euclidean distances in order to avoid noise from distant tissue regions. In Figure 4 we show that the method correctly detects a gradient of expression of two genes. The gene producing myelin and the gene producing the proteolipid protein 1 are highly expressed in the white matter region, but expression is reduced as one gets closer to the gray matter interface. This is expected as oligodendrocytes, which produce these proteins to support other brain cells, are highly abundant in the white matter. Furthermore, our method detected a gradient of expression for the mitochondrial import receptor gene. The gene was expressed at the white-gray matter interfaces of samples 1 and 3, but a gradient of expression in the white matter region was observed in sample 2. This is shown in Figure 5. Sample 2 came from an older individual than samples 1 and 3. Interestingly, this gene has been implicated in late onset of neurodegenerative diseases. Finally, we used the Spearman's correlation values to rank the genes during a gene set enrichment analysis. The gene set enrichment analysis indicated that genes involved in Alzheimer's, Parkinson's, and Huntington's diseases show gradients of expression in the white-gray matter interface. This result provides a landmark to look for drug targets for therapies. We are implementing this new method as part of our analytical suite, Spatial GE. Spatial GE enables analysis of tissue gene expression for spatial transcriptomics data. A QR code to the GitHub repository is at the bottom right corner of the poster. Thanks again for your attention. Please, let me know of questions and comments.